Okay, so we have talked about Mendelian process and transmission genetics. We've talked about the principle of segregation, principle of independent assortment, and dominant recessiveness as the primary uh, type of mode of inheritance that we had when one allele dominates over the other. Now, when we do that, we're really talking about the way the alleles interact, right? You get one piece of genetic information from one parent, one piece from the other. And we're thinking that this piece of genetic code dominates over this piece of genetic code. Or you get two common ones that are the same, they respond as a homozygote. Now, all alleles interact, right, in some technical way, and we'll try to cover how that works. But what we want to do in this couple of lectures here is talk about some other modes of inheritance that apply. Okay. Now, for us, the basic Mendelian rules don't change. They will never going to change. Segregation is going to work. Okay. For right now, independent assortment is going to work. When we get the linkage, we'll talk about some variation of that. But right now, segregation, assortment all works. What's going to happen is we're going to change the phenotypic expression. So really, I like to use sort of silly examples to take us out of technical terms and kind of put something in your head that might be something else. So far, we've started a cake, right? Segregation, the principles that occur there, all these crosses, Mendel's Big Six, are really just how to bake a cake. And what we're trying to do now is we're going to start putting layers on that, maybe put a little icing on that cake. And ultimately, we want to pile those things together and make very nice structures, right? I don't think in this course we're going to quite get to a wedding cake, but we're certainly going to get some really beautiful examples of how to pile on these various rules and these modes of inheritance. All right, so just to remind you, if we go back to Mendel's Big Six here, okay, the crosses that you make determines the genotypes, right? You make a cross and you end up with the expected genotypic system. Under Mendel, under very basic Mendel genetics, dominant recessiveness, you have an expectation of a certain type of ratio that you're going to get, right? Well, all we're going to do now is change this part, okay? We're going to leave the crosses the same 6 crosses. You don't need to learn any more. The genotypes they produce exactly the same, but now the phenotypes and the ratios of how these things group together may in fact change, okay? The relationship between the genotype and the phenotype is based on the mode of inheritance. We've only had one mode of inheritance so far, and that was dominant recessive. Now we're going to try other modes of inheritance. We're going to layer on more complicated things. But very importantly, if you know the Mendel's Big Six, and you know what kind of genotypes you're supposed to get, this won't be difficult. All we're trying to learn is a little bit about how to take these numbers and turn them into a different ratio over here. Okay? If you don't know that, if you're struggling with that after a couple of examples here, maybe you should go back and work some of those earlier problems that deal simply with Mendelian ratios. Okay? All right, so where's our example? Snapdragons, good example, okay, of where we're going to go here. We're going to try an alternate mode of inheritance. If we have pure breeding snapdragons, whites, these guys back here will breed true, all right? And so they're homozygous for the white allele, or little r's is called here. The pretty red ones will breed true as well, okay? And you have a Mendelian expected that when you cross those, you'll get either all red or all white, right? If it follows a simple dominant system. It doesn't. In fact, these plants have been very important for numerous studies. What we see is that these snapdragons follow, at least the color in them, follows a different mode of inheritance than one we've seen before. And this is called incomplete dominance. Incomplete dominance is when the heterozygous condition, and from now on, heterozygotes are going to be critical because that's where we're going to be able to see the particular mode of inheritance that we have. Incomplete dominance is when the expression in the heterozygous phenotype is distinct from either parent. It's generally in the middle. Okay, we do have these things called overdominance and things where you might take a gray thing and a true breeding gray and true breeding white and you get black or something. But for our purposes, we can think of it as being intermediate to it. Okay, so we're going to have these intermediate forms. What that means in incomplete dominance is, unlike Mendel, when we crossed a red and a white, we got either red or white down here, exactly like the parentals, right? Here we don't. We get an intermediate. In this case, we get pink, right? So we have these. The heterozygotes are intermediate between these two forms. Now, if we go to the F2, if we cross two hets, we actually recover some of our reds and our whites as long as having some of these pinks. We end up with what is basically some whites, some reds, and some pinks. And we see that in a very important ratio, okay? Here's a nice little picture showing the red ones, the white ones, and the pink ones themselves, okay? What's important is we end up with a 1:2 to 1 segregation ratio. In our case, the genotypic ratio is the phenotypic ratio. It's actually a lot easier than Mendel than dominant recessive, right? Because we get a 1:2 to 1 expected, and that's what we see. We can exactly tell. Very importantly, unlike dominant systems where we have big A blank, where we cross a het times het and we get 3:1, say, red to white. Here, we actually know, once we look at the color, we immediately know the genotype, right? With a dominant system, you don't necessarily know that. You have to do test crosses and all that because it could be big A, or it could be big A, little a, right? Well, here, what we have is we have exactly a relationship between the color being the red alleles, homozygous for that, the white being homozygous for the white allele, and pink are heterozygotes. So the genotypic ratio is the phenotypic ratio for incomplete dominance. I thought I'd give you a couple other simple examples just so you know that it's not uncommon. Frizzled fowl is a fairly common example. In bird feathers, these inarticulating parts between the feathers, in order to give them a smooth structure for flight, fold out together. Okay, and this might be a chicken that's kind of got that, right? You see the feathering here is fairly, they can lay down and they can form sort of a cover. There are protein level effects that will affect whether these are straight or not, these little articulations here. And if they're not, then you get what are known as fuzzy or frizzled fowl look, and that looks something like this. Okay, now these frizzled fowl are actually very valuable for show chickens because they have this sort of, looks like a bad hair day. Not a particularly good example of hair, but it looks like you've had sort of that, right? Well, these are true breeding. You can take these frizzled fowl and they will breed true. You can take these normal or non-frizzled and they'll breed true. You cross a frizzled with a normal, which is often done for show animals, and you get a mildly frizzled. You get something that's in between this sort of shape that looks like this and something that looks like this, and that's the heterozygotes. Incomplete dominance is somewhere in the middle. We see the same thing for like Siamese fighting fish. These beautiful blue ones are very valued. They are the heterozygotes. And what a lot of breeders will do is keep around a steel blue and a green, and they'll mate those together to assure that all their progeny are going to be these perfect blue like this. Radishes often come in long, round, and oval. The round kind that you often see in stores and places because they're easy to process and roll and get the dirt off and stuff are actually the heterozygotes. If you go out and you buy seeds for the round farm, and they'll be heterozygous seeds, and you can plant them and you can actually get. But if you wait a generation and turn around and try to replant some of those, you're actually going to end up with some long ones and some oval ones and some round ones, and you're going to get all kinds of things. And then it even occurs in mammals, right? There's a behavioral system where we have normal behavior. We have what are known as wobbly mice. These are mice that kind of move around. So we're talking about a behavioral trait here. And the mutation that drives that is actually a fairly simple incomplete dominance mutation. If you take a normal mouse and you breed it with a wobbly, you'll get mildly wobbly. It's kind of an interesting study because this is obviously affecting the neural system. So these are research animals that people can kind of look at and say, okay, what causes this? What protein is malfunctioning or what's building up? And these relate to things like Alzheimer's and other dementia that they'll use these wobbly mice for. But the point is, it's a fairly simple system in this case. Other behaviors are not. Certainly Alzheimer's is not a simple system, but you can actually get some basic information about how the neural system works from these studies, right? So incomplete dominance. Why don't we take that and let's talk about a problem or so with that, okay? And then, so let's do a problem with incomplete dominance. All right, so here's a real system, okay? We have cats, and if you've ever looked at cats, they have nice long tails, okay? My two cats have nice long tails. They can have medium-sized tails, or what are known as short, or they can be minx-like and have no tails like this. for some of the systems, not all the cat systems, but for some of the systems, turns out to be a fairly straightforward system, right? So what is the mode of inheritance here? Well, if we read this paragraph up here, it basically tells us without saying the mode of inheritance. Remember some of those early problems we did with the Mendelian system? We said, oh, by the way, so-and-so, this is dominant over that. Red's dominant over blue or something, right? Well, here you're not told whether you're dominant or not, but you are given a lot of information that lets you figure it out. First off, it says it's at a single locus. So you know you're dealing with a monohybrid system, a single locus trait, which makes it nice. You just need two alleles to do something. We're not dealing with a dihybrid or anything. And then it says the homozygous ones are either ones with no tails or some with long tails, right? That's the homozygous. And then if you have one long tail allele, but you end up with a one no tail allele, instead of long tail dominating over no or no dominating over this, you actually end up with this intermediate called short. So the mode of inheritance obviously is incomplete dominance, which makes sense since that's the point of this lecture, but this is how you would know if you're working the problem. If we're to write this down, we say, okay, long and no, we just use maybe L for long and N for no. If they're homozygotes, you get long tails. Homozygous gives you long, no tail, little n's give you that. Okay, and then what this other sentence says is that if you're going to be LN, you're going to be short. All right, so we know the mode of inheritance basically by reading this, and we can set up this little chart to tell us what we expect to see. All right, well, you can imagine that this isn't going to end the problem. You're going to have to make a cross, right, of some kind. So this is going to be a cross where we're going to ask you to diagram the cross. Same old thing we've been doing, but now we've changed the mode of inheritance. You need to know the phenotypes, genotypes, and some of the ratios that are involved here. Well, the ratios are going to be fairly simple because you're dealing with a single locus. So you're not going to see any 9, 3, 3, 1s or anything else. You're going to see something fairly straightforward. So what are we asking you to do? We're asking you to take a long-tailed cat and a cat with no tail, take it to the F1 and make F2. Classic parental type of monohybrid cross, right? So just write everything down that you see. We need a long tail, we need a no tail cat, right? We know to produce from our last thoughts up here, to produce a long, it's got to be homozygous long. To have no tail, it's got to be homozygous for no, or the no allele, okay? If this is really confusing you, these L's and N's, I mean, some people it does, and they start, for some reason, just because they see different letters, they start thinking dihybrid somehow, right? They start thinking big A's and little B's and all that. If it really bothers you for right now, just make it big A and little a. It doesn't matter. This is just kind of the way it's set up, right? So whichever way will work for you, that's fine. All right. Now we produce an F1. We're obviously going to get a long allele from this guy and a no tail allele from this guy. That's what's going to end up in here. You're going to get heterozygotes. That's going to be big A, little a, if we're using this nomenclature. And these are going to be shorts, right? Okay. It says go to the F2. So if we do that, we have to cross some litter mates from here that are short with some other heterozygous shorts. Not a big problem. Big A, little a times big A, little a. We know from Mendel's Big Six, and I hope you have you in front of you, right? I have mine here. I always do when I'm working problems. So we're down here crossing this, and we're going to get this expectation right here, right? We're going to get a 1 to 2 to 1 genotypic expectation. Well, because we know we're dealing with incomplete dominance, that's going to be the same as the phenotypic expectation. In other words, we're going to get one long, we're going to get one no tail, and we're going to get expectation of 2 short tail cats out of this. Simple problem. We really didn't change any of the underlying genetics. That's my piece of, how to bake a cake. All we did was once we got down here, we looked to see whether those were long or whether they were long and something else. No, this is long, this is no, and then the heterozygotes happen to be short. Okay, change the thing. All right, so let's move from that kind of back toward the molecular stuff just for a moment. Okay, how does all this occur is a big question. At the molecular level, input basically eventually determines this phenotypic mode, okay? Why is that? Because I've point this out, and I'll say it many times. There are no genes for noses, right? There are no genes for ears or eyes or anything else. What there are is there are genetic information that contribute to biochemical pathways, right? They contribute to those biochemical pathways either as enzymes or as substrates or something, but they're part of a biochemical pathway. You make a nose out of many things: skin cells, colloidal cells, cartilage cells, all sorts of things, right? So what's happening is that the alleles are contributing to this pathway. Whatever happens converting a substrate to some eventual product, and it may be many steps, this is where we get this product out here is protein in some form or something, and that's what gives us phenotype, okay? So let's look at a couple of these examples. Let's take a system where we've got, these are the homologs that you would have. This is my locus for some locus, and I ended up with exactly the same genetic information from mom that I got from dad. So I became a homozygote, right? Both of these contribute. So using their color product here, maybe we started out with white. These, that's the substrate. These enzymatically contribute in some way or add product in some way. So maybe we end up with red, something like this, a bright, pretty red, like that. Okay. All right. Let's make it a heterozygote. So see what I did? I changed this allele to another form. Okay. If those contribute and you still get red, like this, okay, then what you've done is what's called haplosufficiency. Haplo is half, and that's sufficient to do something. In this case, it's sufficient to produce enough functional protein here that we still get red. We didn't get anything halfway, we didn't get anything in the middle, we get a normal product, all right? So that's exactly what we get. In that case, this big A allele is called a dominant. That's how dominance works, right? That's exactly how dominance might work, all right? So what's happening here when we don't have a dominant like this, when we have this incompletely dominant? Well, same combination of genetic information. We've got one form over here, and we've got a mutated form here, or vice versa. One of them's the mutant of the other. But you've got two slightly different forms of the gene. Okay, they both contribute to this pathway. But now what we have is what's known as haploinsufficiency. One of these is not sufficient. One of these pieces of genetic code and its product that it's producing is not sufficient to completely drive you from the substrate over to this final product that might be red, okay? It's just not enough protein or it's not enough function. So what do you end up with? Something in the middle, right? That's the simplest way to think about incomplete dominance, at least how it's produced, is that both of these alleles are contributing, but now they can't contribute enough. And it may be that this one's not contributing anything, right? It's just it's just simply not enough product there, or we may have something else. This is known as haploid insufficiency, right? An example might be, okay, big A produces some X number of units of protein per hour or per time or total, and A produces none of these, right? That maybe we could work with that. So let's see what that might give. If big A produces 10 units and little a produces no units, if you got a homozygote, you're getting 20 units. That might give you full color, right? Okay. If you have a heterozygote, in this case, you don't get anything from the little a allele, but you get the same dosage from the big A. You might end up with something like pink, like that. And then if you got the other one producing none, then you can't change anything. You don't add any color, so you end up going back and retaining the white in this case. So this is one of the ways that we actually think that we get this incomplete dominance effect is by what's called haploinsufficiency. You just don't get enough product out of one or the other. And we see this in lots of things, including some genetic diseases that we'll talk about and lots of other things. Other times we see haplosufficiency, which means that it only takes one good copy to completely drive you toward red or toward wherever you're going to go. Okay, the only time you're going to get white is if you completely don't have a big A. Right. Okay, so that's moved us through our first sort of alternate mode of inheritance here, which is incomplete dominance. The next set, the lecture in this set, when we talk about allelic interactions, we'll talk about yet another new mode of inheritance that will be very important to us. It's very similar, but has sort of a different development, different outcome to it.